Thank you very much, Jonathan, for that nice introduction. As Jonathan mentioned, my name is Michael Anderson from the MRC Cognition and Brain Sciences Unit at the University of Cambridge. And for those of you who don't know me, it should become immediately apparent that despite being at the University of Cambridge, I'm not in fact British. I'm American. A state of affairs, by the way, that my British colleagues would, I would describe their approach to this over the last 16 years as one of bemused toleration. Kind of a Ted Lasso situation without the mustache. For these reasons, I'm very pleased to come back to the United States from time to time to get in touch with my roots again. We should be able to hear it, as you said, about our vote. And so I'd like to express my gratitude to the Sage Center for the study here at UCSB Psychology, and especially Jonathan Stewart and Alexis Clubb, without whom none of this would have been possible. Thank you very much to both of you for the hard effort you put in bringing to this. As Jonathan mentioned, the title of my talk is Adaptive Preparing How Control Shapes Memory and Ability. I'd like you to imagine for a second that this has happened to you. You're speeding down a country road. You're going around a bend. An animal jumps out of the bushes and gets in front of your car. You swerve to miss it. You lose control of your car, and the car goes tumbling into a ditch. Fortunately for you, you escape unscathed, except for the fact, of course, that you're seriously traumatized by this occurrence. Fast forward a couple months later, and your arm catches this on the street. Now, if you're like most people, that happens to you because you will be very quickly reminded that associated event. But if you're like most people, you will equally quickly exclude that unwelcome reminding from the awareness. Regain your footing in your mental landscape, go about whatever it is that you're doing, and hopefully, achieve some stability in a short window of time. And if you've ever had something like this happen to you, you have had a close encounter. You've had a close encounter with a process that I refer to as retrieval stopping. In a retrieval stopping situation, you're confronted with a stimulus in the physical environment that's strongly associated to an experience in long-term memory, such that when you see that stimulus, it tends to automatically and reflexively remind you of the associated content. And the question is, to what extent can you focus your full attention on that reminder, stare it right in the face, but nevertheless willfully prevent the associated content from entering consciousness? Can you stop the retrieval process? Can you shut it down? How do you do it? What are the mechanisms that you engage? What are the consequences of engaging those mechanisms for the content that you're excluding from consciousness? And of course, for the broader well-being of the individual. I have been transfixed with this set of questions now for more than 2 decades, embarrassingly. It's embarrassing to admit. And the reason is that I attribute to this set of questions a lot of theoretical as well as practical importance. Theoretically, why is this theoretically important? It's the essence of how we control consciousness. Your ability to think about the things that we do want to think about, and to not think about the things that we don't want to think about. And if we can figure out how retrieval stopping happens, we'll make some, I believe, important contribution to understanding those basic questions about the goal-directed control of consciousness and of behavior. But on a more practical basis, this situation just keeps coming up again and again and again and again. So let me illustrate, right? I've already told you about PTSD, right? So stimuli in the world can elicit a distressing, intrusive memory. But that's not the only situation. And also, in the case of anxiety, stimuli in the world can elicit fearful imagery or thoughts about the future, which are characteristic of pathological worry in that condition. Stimuli, in the case of depression, stimuli can elicit simulations that are typical of depressive rumination in that condition. In the case of addiction, stimuli or context elicit substance use thoughts, which is very typical of addiction. And in the case of OCD, stimuli can elicit contamination imagery, which generates obsessive thinking and obsessive behavior in that disorder. In each of these cases, there's a stimulus that's eliciting content in memory. That content is deeply distressing. The person has severe difficulty in controlling that content and making it go away, whereas the rest of us find it relatively more easy to make it go away. So we think that if we can understand what's going on here in healthy individuals to a great depth, It might provide some insight into what's going awry in these psychiatric conditions and provide a pathway for intervening and helping to reduce. So if our assumption is that this process works by reducing intrusions, but also by diminishing the accessibility of the suppressed content in memory. And to the extent that diminished accessibility, when it has gone awry, when you can't do it, generates this range of psychopathology, these severe distressing symptoms, it really suggests that this process is doing something pretty useful for you. And if it's useful, then I think you refer to the concept of adaptive forgetting, forgetting that serves a functional purpose for the person. It is useful. And my goal today is to talk to you about what I've learned about adaptive forgetting and its potential role in mental health. I'll start off by talking about mechanisms. And over the last 2 decades, we've learned a great deal about how retrieval stopping occurs in the brain, the precise machinery that enables that to happen, and that causes memory to be adapted. I'll give you a little bit of a tour through that. And I'll also tell you about some of the new tools that we've developed to bring that understanding to an ever more sophisticated levels of neurobiology. Then I'm going to change channels and talk about the mental health part, talk about the broad issue of, okay, so we know a lot about how retrieval stopping and thought suppression happens. Is it a good idea? Should we be doing that? Should we be trying to get people to be better at it? And if not, what's the point of understanding it in such great depth if it's not going to be put to some use? I'm going to tell you about an intervention that we developed that causally tests the role of that process in mental health. And then finally, based on these two, I'm going to tell you about my vision for the future, how we're going to marry this deep neurobiological understanding on the one hand with this intervention technique on the other to revolutionize the treatment of intrusive thinking across the same way. Now, to study adaptive forgetting, we need a way of manipulating and measuring retrieval stopping. Thus was born the think no think paradigm that Jonathan was just telling you about. What do you do in the think no think paradigm? It's really, the essence of it is really quite simple. In the think no think paradigm, sometimes we'll give you a cue, something that you learned in the laboratory. We ask you to think about the thing that goes with it. You retrieve the associated content, bringing it to awareness, think about it for a while until the cue goes off the screen. Those are the think trials. The no think trials, on the other hand, we present the reminder and we tell you, don't think about the thing that goes with it. Don't look away, look at the reminder, stare right at it. Don't remove your eyes, don't distract yourself by thinking about lunch or something like that. Just look at the cue and prevent the associate from entering consciousness. And those are the no think trials. And that is the essence of the think no think test in a nutshell. But for those of you in this room wanting more specifics, here's a specific example. So if you were in this experiment, you would be drilled on several dozen paired associates like Ordeal, Roach, Steam Train, and those until you knew those connections fairly well. So that's establishing these associations between reminders and memories. Having achieved that, then you enter the critical think no think. Is there something? The critical think no.
If you can move your lapel mic a little closer to your, yeah, like maybe put.
Even a little on the lapel, the very top.
Yeah.
The very top.
Is it not?
Yeah, there we go.
Okay.
So.
Yes, my students often tell me this. Sorry about that. Yes. So in the think no think phase, you receive 2 kinds of trials. You have think trials on one hand, in which the reminder appears in green, and that's your signal to go ahead and retrieve the associated content and keep it in awareness for the full duration of the trial. But on the no think trials, the reminder appears in red, and that's your signal to stop the retrieval process. So you look at the reminder, you don't look away, you don't distract yourself, but you prevent the associate from entering consciousness. And trials of these 2 types are intermixed in this phase, and the given item is either suppressed or it's retrieved quite frequently in this phase, so upwards of a dozen times, because we want to maximize whatever effects this has on the content that you're receiving consciousness. And then afterwards, we just test people's memory. We either test them with the original reminders that you will see in print, or we can test them with a novel independent cue like insect for Roach. And the purpose of that independent cue is just to see whether these effects generalize across multiple ways of testing the same content. And that has various theoretical implications we'll get to in a moment. So the critical question is, what happens to things that you suppress, the things that you've kept out of consciousness on that final test? How do you decide what's happened to them? By comparing performance on baseline items, at the bottom here, which you were trained on initially, but which didn't appear in this intervening phase, providing you an estimate of how memorable those items would be had you done nothing to them in the interim. So what happens to things that you've suppressed? Just to illustrate, we have other variants of this task that involve different stimuli. This is one involving object-scene associations where the scenes are unpleasant. We have a bunch of different variations with different kinds of stimuli. We've even used autobiographical memories. Generally, the phenomena looks similar across various materials types. And so the general finding that we observe is the more often you suppress this association, you suppress the retrieval of that content, the worse your memory becomes later on. And because the act of suppressing is causing forgetting, we refer to suppression-induced forgetting, often abbreviated to SIF, a SIF amongst friends. And this just tells you it gives you an example of what it looks like. So this is performance on the final memory test at the end of the think nothing task. This is a percentage of items correctly recalled. This is 1,650 people. By the way, this is like a number of years ago. We just took everything that we have either published or didn't publish, pictures or words, negative, neutral, just threw it into one big soup. And that's what this is. In the thinking condition, people recall items very well, much better than performance of the baseline items, which absolutely nobody in this room will write home to your mother to tell them about. But the more interesting thing is that the no-think items are recalled significantly worse than this. So keeping this out of consciousness consistently impaired people's retention on the later test. Striking effect if you think about it, because to achieve that, they had to see the reminders upwards of a dozen times in the interim. In the baseline condition, they didn't even see the reminders. So seeing a reminder a dozen times and trying to exclude it from consciousness is bad for memory. So it engages a process that impairs memory. This exhibits a number of properties that are worth highlighting. Cue independence. What does that mean? That basically means, yeah, you're worse at recalling this picture if I gave you that cue, but you're also worse if I test that picture with a different cue, totally unrelated to that one. So the picture is just generally less accessible, irrespective of the cue. And what that tells you is it's not this association that's damaged, and it's not associative interference from this cue to distracting thoughts, let's say. It's something is happening to the memory that makes it less accessible. It shows up on implicit memory tests. So if I gave you this picture in visual noise so that you couldn't identify what it was, and I gradually made it clearer, it would be harder for you to identify that picture in visual noise if you've suppressed it previously. So you're not even trying to remember anything, you're just trying to say what the picture is, and you're worse. And similarly, conceptually driven implicit memory tests show the same thing. So a variety of different implicit tests show that. And what that I think the implication of that is that this idea that you're banishing something into the unconscious, but it's still out there lurking and influencing you in various ways that Freud had initially proposed. Not so much, at least in this preparation, in this paradigm. It reduces implicit effects as well. And then affect reduction. If it's something unpleasant that you're suppressing, there is muted affect for it, becomes less negative. And that's as measured with skin conductance response or heart rate deceleration, ratings of arousal. Ratings of valence are a little flakier. Sometimes you get the effect, sometimes you don't. But generally speaking, suppressing something makes it a little less upsetting later. Suggesting it has an affect regulatory function. Now, the 1,650 participants might seem impressive, but you might think to yourself, did you just tell me that those are all from your laboratory? Maybe a little suspicious, maybe that's biased in some way because it's not from your laboratory, maybe. But let me assure you that this has been replicated across the world. But in this era of the reproducibility crisis, it's really, I think the gold standard for reproducibility is the pre-registered replication work, right? Well, one is on its way. Jonathan Fawcett and Benjamin Levy, without me, it was an independent effort, agreed with Nature Human Behavior to do a pre-registered replication of this phenomenon and to publish it irrespective of how it turned out. They had 60 sites in 19 countries in 12 languages across 4 continents, over 3,000 participants. And they've allowed me to tell you that the smallest effect that they observed of the various effects that they're testing had a base factor of just shy of 2 billion. So it's on solid footing. This is the lesson. The other important development is that a former postdoctoral fellow in my lab, Davide Nardo, did the field a real service. He published a best practices, kind of a hitchhiker's guide to the galaxy kind of review paper, going into intricate detail about how to best conduct experiments like this, design decisions, things that, the insider knowledge that we have in the laboratory about how to best do it is insider. It's all there in the paper, and you can go read it yourself. But importantly, he included a training package, and if you knew what went into this training package, you would be seriously impressed. But what it provides is MATLAB code, subject instructions, experimenter scripts detailing every move the experimenter makes, Excel spreadsheets, multiple training videos, one by me, a lecture on how to best do this stuff, and an HTML that links it all together into a structured training so that anybody in this room, quite apart from your advisor, if the advisor doesn't want to have much input, you can just go do this, download it, and do it without even consulting us, though we'd be happy to consult. And if you're interested, there it is. You can just, there's a QR code that you can use to download it. We were delighted to learn, we, as in me, about 10 days ago, I was delighted to learn that in the 2 years since this has been published, this package has been downloaded 13,000 times. And so it really suggests that people are interested in adaptive forgetting. And between that fact and the fact that there are 60 sites across the world now that are up and running and doing this kind of work, I think you can expect in the coming years that scientific progress on this phenomenon will increase, perhaps dramatically. So with that paradigm and that phenomenon, then our goal was to try to understand, to establish a systems level model of how adaptive forgetting happens. And what I've done here is I've provided the broad outlines of such a systems level model in terms of data that I've taken from a paper with John Gabrielli in 2004. So what we found is that when people suppressed the retrieval process, they engaged the right dorsolateral and ventrolateral prefrontal cortices. The more they engaged that, the bigger the suppression-induced forgetting was. And at the same time, they significantly reduced activation in the hippocampus bilaterally. And on the basis of that general pattern of findings, John and I proposed that the way that people stop the retrieval process and the mechanism that causes forgetting is the engagement of a right prefrontal control mechanism that suppressed hippocampal activity to interrupt the retrieval process and to disrupt retention. And that broad outline is the general set of concepts that we still work with today, though, as you'll see, we've made great progress in elaborating beyond that growth. The first thing to be established was using dynamic causal modeling, which is an effective connectivity analysis method. We can repeatedly confirm that the prefrontal cortex does have a top-down causal influence on hippocampal activity and a negative inhibitory modulation effect. So it's a widely replicated phenomenon. And we've also established that there are other modulatory elements as well. So when you're suppressing something distressing, we get parallel modulation of the amygdala and the hippocampus. So that would account for some of the affect modulatory effects that I just told you about. And in addition, we also see neocortical modulation along with hippocampal modulation. And so, for example, and this really depends on what it is you're suppressing. If you're suppressing a scene, you get reduced activity in the parahippocampal place area together with the hippocampus. But if you're suppressing a visual object, you get parallel modulation of the hippocampus and fusiform cortical regions involved in conscious object perception. So the cortical regions are really content specific. And to summarize that, we refer to the reinstatement principle. which governs which cortical area it shows the modulation. It basically means whatever area of the cortex would show reinstatement if you were to retrieve that content are the things that are the areas that are modulated by top-down control during suppression. So that's the reinstatement. Yeah, okay, so there are these other targets, but in my presentation today, I'm going to focus on the frontohippocampal circuit. And that's been a major focus of the laboratory, trying to elaborate what's going on there. In fact, the broad, long-range goal of our program is to develop a multi-level integrated model of adaptive forgetting, this frontohippocampal model specifically. That model would explain the cognitive mechanisms that underlie retrieval stopping and their impact on behavioral metrics of memory, whilst also specifying the brain systems that underpin this retrieval stopping ability, systems that initiate a top-down inhibitory control signal that modulates hippocampal activity, as well as the local circuit mechanisms by which the hippocampal activity can be modulated. And I'm going to tell you a little bit about that today. And finally, the cellular and molecular mechanisms that enable these local circuit mechanisms to gradually tune the plasticity and induce adaptive forgetting. We have worked at all of these levels of analysis, work our hypotheses at all of these levels of analysis. And the goal is the full vertical integration from the mental to the synaptic. In today's presentation, I'm going to focus on these levels of analysis, but you might be wondering, how are you going to do that? Let me tell you. We spent 7 years developing an animal model of memory inhibition using rats. And it works fabulously. It works a treat, as they say in English. So our first paper on this was in 2018. There was like 7 experiments in it. We went through and showed systematically that rats show all the functional properties that humans do. So it is a species general, at least within the mammals, it is a species general set of mechanisms and abilities. And what this does is it opens the door to the full armamentarium of methods in neurobiology, lesions, optogenetics, chemogenetics, molecular biology. And especially if we can now transport this into a mouse model, then things really start to get exciting. So we can, what that enables is causal mechanistic tests of the precise circuits that suppress it. Turn it on, turn it off, for optogenetics and chemogenetics. Precise specification of the inhibitory interneurons within the hippocampus that achieve this kind of modulation. So it's the level of specificity that is just hard to imagine given the set of questions that you're thinking. Okay, so if you're interested, I invite you to check out our Nature Reviews Neuroscience paper. This is with S. Subbulakshmi, a former PhD student of mine who's now a postdoc at Stanford. Or if you're interested in the animal model, check out the Nature Communications paper, which is done with a molecular biologist by the name of Pedro Bekenstein. Really lovely work, and there have been papers since then. Okay, so part 1 then is talking about retrieval stopping, the retrieval stopping mechanism, trying to elaborate this schema that I presented to you at the very beginning. What do we know about that? What does it mean? What have we discovered here? And I'm going to argue that what we've discovered is the existence of a frontotemporal inhibitory control pathway. Everybody knows about inhibitory control, but most people study inhibitory control in the form of response inhibition, stopping actions. This is different. I'm going to argue that it's overlapping. There are domain general aspects to this pathway, but there are unique aspects of it to controlling unwanted thoughts that are crucial. So in part 1, I'll tell you about the evidence for domain general control. Part 2, I'll tell you about the property of dynamic targeting that enables the domain generality. And then I'll talk about evidence that what you're observing here genuinely is hippocampal inhibition. Qualifying this as an inhibitory control mechanism. And then I'm going to give you a sneak peek into the circuit level of analysis that I was alluding to earlier, talking about the medial septal pacemaker suppression hypothesis, as well as the thalamo-hippocampal modulation hypothesis, which builds on the nucleus reunions of the hippocampus. Okay. Everybody has heard of the idea of inhibitory controls. And the idea that it might be a domain general way of controlling behavior. Certainly, anybody who's ever listened to Adam Aron, people know who Adam Aron is, like a handful of you.
I was in lab manager.
Yours like, anybody who's ever listened to Adam Aron would take it for granted that there exists a domain general control mechanism. But this is a very strong claim. And that's sort of funny that the very same thing that's helping you answer things in your world, physical action, may be the same mechanisms that help you to not think distracting or distressing thoughts or feel distressing emotions. Is there really one ring to rule them all? Is there really one mechanism that suppresses all of that kind of content? It's a pretty bold claim. And so we asked whether or not this might be reflective of such a domain of inhibitory control. We asked the question, is stopping a thought similar to stopping an action? Similar in what way? Similar in the brain regions that they engage, but also similar in the precise pattern of activity within those brain regions. So is there any evidence that that's the case? Our approach to this was just to look for a within subjects conjunction. So brain regions that are more active when you're trying to stop a thought, no think greater than think, and brain regions that are more active when you're stopping an action, stop greater than go. And so if you were in one of our experiment, or in this experiment here, you'd be multitasking. So do these little blocks of like 40 seconds where you're stopping actions, like stopping key presses. On the one hand, that's the stop signal task. But in the other mini blocks, you're stopping thoughts, you're stopping the retrieval process. And so we're just going to look, the prediction is if you do the conjunction analysis of those two, there will be brain regions that do both. And that may be more likely to reflect a domain mechanism. You do that, and this is what you get. And it's actually a very clean story. This part here, the right ventrolateral prefrontal cortex, would make Adam Aron very, very clean, because that is the region that he has historically emphasized as being the seat of domain general inhibitory control. But in addition to that, one observes the right anterior dorsolateral, which is the region I've emphasized over the years as being the seat of domain. I'm very pleased with that. But you get both. And you get this right supramaxillary gyrus. You also get, it's not shown here, but you get anterior cingulate cortex as well. Very strongly right lateralized and consistent with the literature on inhibitory control. Now, we were a little worried that maybe the similarity of these two forms of stopping, maybe it has something to do with the way that we set the experiment up. Maybe it happens because people are going back and forth and back and forth and back and forth between stopping actions and stopping thoughts. And maybe when you do that, they just get more similar to each other. There's a carryover effect of some sort. So what did we do? We did a meta-analytic conjunction analysis. We went out and we got every stop signal study that had ever been published and every think-no-think test that had ever been published, did meta-analyses for each of them separately, then did the conjunction of the two to see whether or not you got the same answer. And that's what you get in blue. It's the same. It's the same despite the fact that these are independent data from multiple labs across the world using massively different versions of these tasks. And yet it couldn't be more similar to our within subjects conjunction. And this really makes me confident that what we're observing here is a reliable, robust set of regions that seem to be engaged by both of these forms of stopping. Now, this is for the very most you, which is me. This is the Brodmann's regions. So you get this area straddling BA44 and 45. But in this, you get this right anterior DLPFC region, which straddles this tri-state area here between 946 and 10. So we call that anterior DLPFC, ADLPFC, not the whole DLPFC, the anterior portion, and that's important. So both of these regions seem to be important to achieve this function. And you are fairly in that. So the key findings are that we get shared stopping regions. It's both the VLPFC and the VLPFC. So anybody who wants to claim that it's one and not the other, the data just don't agree. That's all the data. That's not my data, it's all the data. And this is, in addition to looking at the univariate signal, we looked at cross-task decoding. So maybe just because they're in the same region doesn't mean that it's the same function. But it could be that there's little segmented parts of these regions that have to do with one function versus the other. So what we did doing cross-task decoding, we looked at pattern of activity across all of the voxels in each of those prefrontal regions. We wanted to see whether we can train a classifier to distinguish action stopping from action going and see whether or not it could tell whether you're trying to suppress your thoughts. And it can. You get this cross-task decoding, and it goes both ways. I can train it on suppressing your thoughts and tell whether you're stopping your actions too. And in fact, in the think, no think phase, you look at blocks, successive blocks as people are suppressing and suppressing and suppressing. In the very early blocks, the classification accuracy is upwards of 80%. It's quite accurate. But as you get better at pushing that out of mind, the item gets progressively less accessible, classification accuracy declines, and the slope of that decline across repetitions predicts subsequent forgetting. So I can predict not only whether you're suppressing your thought or not, but whether you're likely to forget later on. That's just by training a classifier on action stopping. So that really suggests that what we're dealing with here is a domain general region. But how is the domain general region achieving domain generality? It's controlling very diverse things. On the one hand, it's controlling physical actions. It's controlling my movement here, stopping that movement. Motor cortical regions involved in that. On the other hand, the hippocampus is involved in episodic retrieval. How is it targeting one region versus the other? It has to have the ability to dynamically alter its communication to other structures to achieve that domain generality. And we refer to that idea as dynamic targeting, the aiming of inhibitory control. And so to look at this, we basically, we wanted to first verify that there are these patterns of modulation within the putative target regions that indicate that inhibitory control is actually happening. So, for example, motor cortical area M1 will be the region that mediates the key press, led them into stopping with the stop signal test. And the hippocampus is the region that we believe would be targeted by mnemonic suppression. So if you look within M1 during response inhibition, you see upregulation during the go trials, and you see downregulation during the stop trials, consistent with inhibitory modulation. In the hippocampus, you see during the think no think test, you see upregulation during think trials and downregulation during no think trials, again consistent with that being a target of control. You get a crossover interaction such that during motor response inhibition, M1 is more suppressed than hippocampus, but during retrieval suppression, hippocampus is more suppressed than M1. This is really consistent with the idea that there's an inhibitory control mechanism which is targeting this thing or that thing in a goal-dependent manner. Can we establish a causal network that shows that? We used effective connectivity analysis, dynamic causal modeling, which included 4 nodes, BLPFC, DLPFC, M1, and hippocampus. And we looked at 72 different models of the ways that these regions could interact with one another. And how can you generate 72 different models? Let me give you a flavor. We could have models in which we pretend that only the VLPFC matters and it modulates the hippocampus and M1, or models in which only ADLPFC matters and it modulates M1 and hippocampus in the context of that way, or both matter and both matter and they talk to each other, or there's bottom-up connections from the hippocampus to the VLPFC or to the DLPFC and so on and so forth. There you can come up with a lot of different variations, and then we fit those models to the brain and data, and one model pops out of all that with a exceedance probability of 0.92. And that's the model in which both of these regions target the hippocampus during retrieval stopping, but not M1. And then during action stopping, both of these regions together target M1, but not the hippocampus to suppress action. And they talk to one another. So they're in concert with one another. There isn't, models with only one of these regions simply don't work. So this really, I think, suggests that is consistent with this idea of a frontotemporal inhibitory control pathway. So yes, these two regions are domain general. They both of those regions seem to be involved in both forms of stopping. but they act ultimately in different ways, that through pathways that modulate motor cortex or pathways that modulate the hippocampus. But you might think to yourself, okay, I believe that there's a distinct frontotemporal pathway, but I'm not so sure about labeling it inhibitory control. Why all the business about inhibition? Prove to me that there's inhibition actually happening here. And so now I want to, so is this structure really being suppressed? And is the only reason I'm saying that is because bold signal is reduced here? That's not necessarily a strong reason, but that modulation of bold signal in the hippocampus shows a set of properties that have led me and others to conclude it reflects an active process of suppressing that structure to interrupt retrieval. Property number one, intrusion dependence. What I mean here is you can ask people after each think and no think trial, did the item pop into consciousness during that trial? Yes or no? And they tell us, yeah, it did. No, it didn't. If you separate the trials according to whether there's an intrusion, you get much more profound downregulation of the hippocampus during intrusions than you do during non-intrusions. The top-down frontal lobe of the hippocampus is larger and more negative during intrusions than during non-intrusions. The correlation of bold signal in the hippocampus during intrusions with forgetting is much greater than it is during non-intrusions. The intrusions seem to be where the action is. So that's consistent with an active regulation. Second property, GABA dependence. So the depth of this dip here is related to individual variations in GABA within the hippocampus. GABA is the chief inhibitory neurotransmitter of the hippocampus. And we can measure it, we measured it in a multimodal imaging study using magnetic resonance spectroscopy and fMRI. And so we can quantify GABA in the hippocampus, the visual cortex, and the prefrontal cortex. We see that only hippocampal GABA, not GABA in those other regions, predicts the depth of this dip. It predicts how much you forget later on. So if you have more GABA, then you're a better forgetter. And it predicts this effective connectivity with the prefrontal cortex, such that if you're low in hippocampal GABA, there is no effective connectivity. So you need the inhibitory boots on the ground in the hippocampus for that prefrontal region to have its effects, to cause the forgetting. So that leads us to believe that it's really an inhibitory effect. Theta bold suppression, property number 3. So it's not just that there's reduced bold signal, there's also evidence for reduced electrophysiological signatures of retrieval. And so here we did a simultaneous EEG fMRI study where we were able to source localize theta activity in the hippocampus and show that during suppression, there was a significant reduction in hippocampal theta, and that correlated with the magnitude of the bold signal reduction, that correlated with the forgetting that people exhibited on the Levy test. And so that basically is indicating that the reduced bold signal is reflective of an interruption of processes that are known to be necessary for retrieval. Finally, and this is my favorite, I love this effect, the amnesic shadow. So if you're suppressing the hippocampus, if you're down-regulating activity in that structure, what does that mean? What does it do? Is it like maybe creating a little lesion in the hippocampus, like a reversible lesion? Is it the hippocampus just worse at everything that it's supposed to be doing? So it's interrupting retrieval, sure, okay, maybe you believe that, but maybe it's also disrupting encoding. Maybe it's also disrupting consolidation. And whatever other functions you might, maybe spatial processing, maybe whatever other functions you attribute to the hippocampus. And so we tested this in the following way. So this is now the think, no think phase of task. If you were in this task, you've just seen pump, which is associated to some other word, and you've tried to suppress the retrieval of this associate, stealing it from consciousness. Well, that trial ends, and according to this analysis, then the hippocampus in the intervening intertrial period should be in a suppressed state. There should be some carryover of its dysfunction. And if that's true, then if I simply stick something else in there for you to make some incidental judgment on, you should remember that less well because you have anterograde amnesia because of the previous suppression period. Similarly, If I present this and then afterwards you suppress something, that should downregulate hippocampal activity. And to the extent that there is a trace now of the peacock in your hippocampus, it now is deprived of the necessary processes for stabilization and consolidation. So you get retrograde amnesia. And if you do both, you should get double the effect. All of those things are true. So that you get the amnesic shadow effect, and it's been repeatedly replicated now with different materials. You can even present a reminder in this period to something you learned previously. So you learn a bunch of pairs before the experiment, and then you just present the cue and remind people of it. And then now they'll forget the thing that they previously encoded as well. So it works for new novel encoding, and it also works for disrupting things that are already learned in memory. So the amnesic shadow really suggests that what we're observing here is a suppression of the functional integrity of the hippocampus, consistent with temporal inhibitory control mechanisms. So I haven't told you anything yet about the pathway by which this happens. And this is kind of an important point for those of you who are anatomically knowledgeable. You might know the somewhat uncomfortable fact that there are no direct projections from the DLPFC to the hippocampus. They don't exist. So how does this work? If it works, it has to be a polysynaptic effect, so multiple steps in the pathway. Moreover, projections from the prefrontal cortex, they're not inhibitory. They're excitatory, they're glutamatergic. So to the extent that there is a suppression of hippocampal activity, it has to be by modulation of local populations of inhibitory interneurons. How does that work? For years, I haven't really known. I've just had to wave my hands and say, look at the effective connectivity analysis. It's happening. But now we're getting more specific than that. We're actually trying to show the circuitry based on known primate and rodent neuroanatomy. And so our first hypothesis is the medial septal pacemaker suppression hypothesis. And I'm willing to bet everyone in this room knows that the hippocampus is important for memory, right? I'm willing to bet that very few people in this room, perhaps no people in this room, know that the ability of the hippocampus to do its job critically relies on a small nucleus in the basal forebrain called the medial septal nucleus. If you lesion the medial septal nucleus in rats, reversibly or permanently, you can render rats amnesic. Their hippocampus is fine. But when you disrupt the medial septal nucleus, it messes things up. Why? Why does such a tiny basal forebrain nucleus have such an outsized effect? Because this structure sends long-range projections to the hippocampus, cholinergic projections and GABAergic projections to the hippocampus, which working together help to achieve the theta oscillatory rhythm, which is necessary for encoding and retrieval. The hippocampus doesn't do its job as well unless its oscillatory activity is entrained in this way. And that enables communication with cortex as well. And I want to call your attention to the long-range GABAergic projections, which have the striking property that they terminate exclusively on GABAergic interneurons in the hippocampus. In other words, these long-range projections land on inhibitory interneurons, and they inhibit them. They inhibit the inhibitors. What does that do? It raises hippocampal activity. Now, thinking this through, if you wanted to raise tonic inhibition in the hippocampus, what would you do? You'd inhibit the inhibitors or the inhibitor. You would inhibit the medial septal nucleus, and in doing so, deprive the GABAergic projections that go here, let the inhibitory interneurons in the hippocampus run free and suppress all of those pyramidal neurons that they project to, and also disrupt the theta activity that's necessary to generate retrieval and encoding. Works with the anatomy beautifully. So is this true? Well, I have a poor postdoctoral fellow, very patient. She went back across 10 data sets, 330 participants, and hand traced the medial septal nucleus in every single one of those people. And she analyzed the data to see whether or not the medial septal nucleus was suppressed. It is. So this is 330 fMRI participants. This is a picture of where it lies, medial septal nucleus, and this is it. Put it on right up so you can see it. But anyway, during the no-think trials, it's significantly suppressed compared to the think condition. And if you look at a subset of participants of 103, for which we have intrusion ratings, trial by trial, that reveals that this downregulation is driven largely by the intrusions and not so much the non-intrusions. So, to put it very intuitively, something pops into consciousness and you say, no, slam on the mental brakes, suppress the medial subthalocus, and you basically create these effects in the hippocampus. And that is how forgetting works. And we have another parallel project with Maite Crespo Garcia, where she's now established that hippocampal theta reductions in the hippocampus using a parametric modulator analysis coupled with medial subthalocus gold signal reduction on a trial by trial basis. So she's able to go through on every individual trial, look at source localized theta activity in the hippocampus, and relate that to suppression of medial septal nucleus activity. So that exactly as predicted by this hypothesis. And she has also shown that the effective connectivity between the DLPC and medial septal nucleus is inhibitory. So that's one hypothesis. The second hypothesis, as if we need another, but hey, we have another. We actually have more than this, but only two. Is that we focus on the nucleus reunions. The nucleus reunions is right there. What is the nucleus reunions? Show of hands, how many people have heard of the nucleus reunions? One, two people out of the whole audience. Nobody in the human community has heard of the nucleus reunions. But if you're somebody who does rat research or animal research in general, you're all over the nucleus reunions, right? Because it has the distinction of being the principal mode of communication between the prefrontal cortex and the hippocampus. And it's located in the ventral midline region of the thalamus. It's like a long cigar-like structure. And recently, a colleague of mine at the University of Glasgow, Mick Craig, has shown that the projections from the nucleus reunions to the hippocampus are primarily targeting inhibitory interneurons, positioning it or being the structure that modulates hippocampal activity to suppress retrieval. So it's an alternative mechanism to what I just described, not mutually exclusive. So this is a collaboration. So this is done by Mahek Kirpalani, PhD student, in collaboration with primate neuroanatomist Helen Barbas and Vasilis Zakopoulos. And yeah, that works too. So this is 35 participants in a very specialized designs that enable us to detect activity in a small structure. And we get significantly increased activity in the no-think condition relative to the think condition, primarily during intrusions relative to non-intrusions. So, and this is the significant development because one of the bodies of work that is being developed in the rodent community that is focused on the nucleus reunions is fear extinction. Stephen Marin, for example, is all over the nucleus reunions as the way in which the mediator of fear extinction. So I've given you a bit of a guided tour of the mechanisms that we've unveiled, and I've made the case that what this naive caricature from my article with John Gabrielli about there being some way of modulating hippocampal activity by the prefrontal cortex has evolved into the specific claim of a frontotemporal inhibitory control circuit, which is composed of domain general components, but this bespoke pathway that targets structures involved in memory. And I've told you a little bit about the circuit mechanisms by which this hippocampal modulation may take place. And now we have access, now that we have our rodent model, we can get in and get causal and about how that's actually working, at least in rats. Okay, so that's mechanisms, but why would we go to the trouble to understand those mechanisms if we don't know whether or not it's any good to do this? So now I'd just like to turn to the question, should we suppress our thoughts? Well, again, Parker, I'm going to tell you a little bit about an intervention that we tried to see whether or not engaging in this behavior, what the effects were on people's mental health. Surprisingly controversial topic. Anybody here who is at all clinically oriented or clinically adjacent probably, or who sees a clinician for that matter, or who has read mental health advice online, will know that the idea of suppressing your thoughts, no. No, we're forbidden. Thou shalt not do that. And the reason, the first view that I'll talk about is the maladaptive coping view, started by none other than Sigmund Freud, who I'm sure you all recognize from this photo. Freud said that there exists a repression process that banishes conflicting content into the unconscious, or distressing content into the unconscious. And it's there. It's not just hanging around. It's doing stuff to us. It's coming out in our dreams. It's coming out in our attitudes. It's coming out in our emotional reactions. It's coming out on unconscious patterns of thought and occasionally back into consciousness as well. And according to Freud, it is this repressed content that is the origin of psychiatric symptoms. And the goal of psychoanalysis is to reveal what has been repressed, to unearth it, so that you can dissipate its energy and to terms with it, and you no longer suffer those symptoms. This collection of ideas has had a dramatic effect on 20th century clinical psychological thought, an effect that was amplified by Jonathan's friend, Dan Wegner. Dan Wegner made the further proposal that suppression isn't even effective, right? If you try to suppress your thoughts, it's intrinsically ineffective. It will cause them to rebound back at you. You'll try to suppress even harder, and they'll rebound even harder. And if you keep going through that cycle, before you know it, you have OCD, you have anxiety disorder, you have depression. And so the original sin is an attempt to suppress thoughts that you don't want to have. This collection of ideas had a dramatic influence on psychological thinking that persists today across multiple sub-areas of clinical psychology. I put here one for your enjoyment. But there are many I could draw from. This is from the Handbook of Anxiety, a chapter on experiential avoidance, a generalization of the concept of thought suppression. Experiential avoidance, or the unwillingness to remain in contact with distressing internal experiences, along with attempts to control or avoid distressing internal experiences, has been associated with a range of psychopathological symptoms of anxiety and fear. These attempts to control internal experiences can exacerbate distress and limit behavior. It's been linked to fear of negative evaluations, worry, fears about cues, fear of the significance and meaning of thoughts, and fear of traumatic events. It is a promising construct of research on transdiagnostic models of anxiety, and it appears to be an important construct to target in treatment. We must avoid avoidance. Thought suppression is avoidance. We must avoid thought suppression, right? Okay. The problem with this is inhibitory control. What does inhibitory control say? Control is good. Not being able to control, bad. So, for example, stopping actions. Avoiding intrusive thoughts, resisting distractions, suppressing urges, resolving linguistic competition, regulating externalizing behaviors. There's theorizing ongoing in clinical psychology and cognitive neuroscience that says that if you have good inhibitory control, you will be better at all of those things. And of course, correspondingly, if you are deficient in inhibitory control, you will be vulnerable to all of those things. These two things can't both be true. They are impossible to reconcile, and nobody seems to have even noticed. Even have people happily go and propose that thought suppression is evil, and then give a lecture on how inhibitory control is good 2 days later, right? So they just don't notice. And let's now to put a fine point on this. I just got done arguing that retrieval stopping is inhibitory control. So now let's plug this in. Retrieval stopping, the unwillingness to remain in contact with distressing internal experiences, check. With attempts to control or avoid distressing internal experiences, check. Has been associated with a range of psychopathological symptoms like anxiety and fear, and you can go on like this. And what's glaringly clear is that that approach predicts that suppressing thoughts should harm your mental health. It should increase anxiety. It should increase depression. It should increase OCD and PTSD. That's why nobody says that you should suppress your thoughts. So they can't both be true. So we tested it. We sought to train people to suppress their thoughts. I want to introduce you to this person, Zulkaida Mamat, former PhD student of mine, now a postdoc at Stanford. And during the pandemic, she couldn't do the brain imaging study she wanted to do. And so she suggested, because everybody was suffering from increased anxiety and depression worldwide, let's try to apply what we know to see whether we can help people, maybe reduce their anxiety and reduce their pain. Let's train them to suppress their anxious thoughts. I'm surprised that my reaction to this was, I was a little nervous because over the years, I've been cowed by clinical psychologists. Emily Holmes had the office next door to me. She took just no opportunity to tell me how ill-advised my program of research was. And she made a dent. And so I was a little hesitant. And Zuli said, don't you believe this? And so I couldn't really get out of it then. So we tried the intervention. And this is what we did. 120 participants, all adults across 16 countries, average age of 27, 77% female. All of this research was done via video conferencing, which enabled us to run 120 patients in 16 countries. So a very diverse sample. What did we do? We wanted, the objective of this study was to look at mental health. We wanted to shift people's mental health from being worse to being better. We're not so concerned about medical system. So as such, we didn't want to suppress neutral word pairs or neutral pictures. We're not like building a mental muscle there. We wanted them to suppress the stuff that was on their minds. And so in days 1 and 2, we elicited 20 fears of the future, 36 neutral events in the future, and 20 hopes. And just to give you a concrete idea of what this looks like, the fears had to be really distressing to them. They had to be cycling through their head, causing them misery. Back to you. So for each one of the fears, they listed a cue, a central detail, and they gave us a tagline for us what it was. So this woman, this is a real example. She was afraid that her dad would get COVID-19 and be hospitalized because he was so vulnerable. And so for her, the cue was hospital. The central detail was breathing. And the tagline was parents get COVID-19. No pictures were presented. That's just to illustrate for you. And the neutral items, going to the optician next week in Fisherton. So the cue would be optician, the detail would be Fisherton. The positive items, watching my sister get married, which was something that we think about the future, the cue would be wedding, the detail. Yes. So then, with those in hand, we gave them 3 days of suppression training. So they attempted to suppress these distressing thoughts 12 times on each day. And then just to see what that looks like. So they would get their title in red if it was a no imagine or no think trial. And their instruction was as follows. Look at this, identify what the fear is, acknowledge it, and then stop yourself from thinking anything else in relation to it. So if an image comes to mind, push it out. If your thoughts start to spiral, shut it down. Turn your attention to the cue. Don't self-distract. Don't think about lunch. Don't move your eyes away. Just don't think. The no imagine trials, the cue was presented in green. We have to think or imagine the associated content. Because we wanted their mental health to improve, we never had them imagine negative stuff. They only ever imagined positive things or neutral things between subjects. And in the no imagine condition, they only ever suppressed negative things or neutral things, again, between subjects. Then we tested their ratings of vividness of the fears, whether they can remember the central details and what their effective ratings were. And then 3 months later, we did the same thing. And then we measured mental health beforehand, immediately afterwards, and at a 3-month follow-up. And we measured mental health with negative mental health indices like state anxiety, worry, negative affect, and depression, positive mental health indices like positive affect and well-being. We computed change scores post minus pre or follow-up minus pre. And we, just for simplicity, we computed a principal components analysis to derive a latent variable which summarized the degree of mental health behavior. And we included predictor variables measured at the outset of the study, including trait anxiety with STI trait and post-traumatic stress, and the impact of event scale pandemic related. And that's because we wanted to know if you're somebody who's super high in trait anxiety or you're experiencing PTSD, how does this intervention influence you? Because maybe you're less good at doing this. Maybe it would even harm you. So first we verified that we get the forgetting effect. So suppressing this distressing content, does it actually induce the forgetting that we're used to seeing in the lab? Yes. So we get suppression-induced forgetting on the recall of their critical details they provided for us. Significant reduction in that. A significant reduction in the perceived vividness of the fears, and significant reduction in affective intensity, perceived affective intensity of the fears. We were concerned that about ironic effects. According to this ironic processing theory, when you try to suppress your thoughts, it should actually make matters worse. You know, trying to suppress a fearful image in the future should make that image more vivid. It should make your anxiety about the image worse. So we looked really hard for evidence of that. What I'm about to show you is all 61 participants who suppressed their fears. What's plotted on the x-axis here is the change in vividness post minus pre. So anything to the left of 0 is a reduction in vividness. Anything to the right of 0 is an increase in vividness. These are every one of our participants, and you don't need statistics here. And these participants are sorted, by the way, according to the initial emotional intensity, from least down here to the greatest emotional intensity of their fears. And you can immediately see that, especially the people who had emotionally intense fears, showed greater reductions in vividness. And that was a modest but significant relation. So it's the opposite of what you might expect. Maybe it's the intense fears would be really hard to suppress, but in fact, there's an anxiety ratings, similar story here. Again, the overwhelming proportion of people showed a reduction in anxiety. If you read the paper, we scoured our data for any evidence of an ironic effect, and we found absolutely none, even when we look at individual people. So how does suppression affect mental health then? So on the y-axis scale, this is our latent variable, make it simple. Negative scores mean improvement in mental health symptoms. You can see that when people suppressed fearful things, they were significantly better in their improvement compared to when they were trained to suppress neutral things. So 60 people in each group. And so what they're nice is that these two groups are matched for what they generated initially. Everybody generated all types of things. They all got suppression training. They all learned to use the method. They all interacted with our charming experimenter Zuli, which surely improved their mood. But the ones that improved in mental health were the ones that suppressed their fears. And this is just to show you that imagining positive does give no mental health benefit of imagining positive content. Here is every dependent measure. This is every data point from every scale, which is anxiety, worry, depression, negative affect, positive affect, well-being, suppressed negative, suppressed neutral, pre, post. The important point is suppressing negative stuff reduced worry, reduced depression, reduced negative affect, increased well-being. Across all these measures, we've got these beneficial effects. There's even a few beneficial effects in the suppressed neutral case, but just less overall. So one final thing, which we paid very careful attention to, was whether or not the most vulnerable of our participants might not have been benefiting as much as the relatively more healthy of our participants. Maybe this benefit is just the people who are basically pretty healthy anyway, and maybe they feel a little bit better now. And maybe the ones that were really suffering from trait anxiety, they don't show any benefit, or maybe even they're a little bit worse. Exactly the opposite was true. The biggest effects were people with the highest trait anxiety and the highest pandemic-related stress, post-traumatic stress, and the highest initial depression. And it's not just regression to mean, which if you look at those correlations, it's the usual condition, they don't occur. And the difference of those correlations is significant. So the people who are the most vulnerable benefit the most. People feel that this is how well did you suppress your fears on day 1 versus day 3 on a 5-point scale? Starts off at 2.7, and by day 3, 4.5 out of 5. Well, almost everybody is saying, yeah, I could do this. 73% of our participants said that they were surprised, they were very surprised at their ability to do this. Nobody saw it coming. They thought, I'm not going to be able to do this. As I'm sure many in the audience right now are probably thinking. They thought that, and then by the end of the experiment, they're like, I can look at that reminder and not think about that. No problem. 87% after 3 months, 87% said it was useful to them. And speaking of the 3-month retention interval, more than 80% of our participants, if you look across here, this is how often do you get to use a pie chart? So they never used it during 3 months, sometimes use it or often. So sometimes and often, more than 80% of our participants said that they continue to use it. And I need to stress here, I didn't tell them to continue to use it. I said, see you in 3 months. That's what Zuli said to them. With no instructions. And 80% continued to use it. The other people with PTSD and depression, an impressive 40% roughly said that they used it frequently during that 3-month period. This is their subjective impression of whether or not this suppression process helped their mental health, either the mood or anxiety. So we asked them, what did suppression do? Did it harm your mood or anxiety? Did it do nothing? Did it improve it? An overwhelming fraction of the people said that it improved both anxiety and mood, especially if you have PTSD or depression, where you get 70 to 80% of the people saying that they experienced improvements that they directly attributed to mental health suppression. And the subjective reports, in their own words, in practice, I couldn't believe how effective it was. It made me realize how powerful my brain is. I always thought just dismissing things, but they worse, brushing it under the carpet. Power is in useful tools, some distressing relevant thoughts. It can definitely help me have more control over anxious responses that randomly appear. It's really powerful to block worry thoughts. Definitely intend to use them again. It's having working with a use strategy is very effective in stopping even thinking negative and disturbing things. We have an online supplement to this where we give the subjective reports to every participant in the negative condition. I invite you to go there. So this gives us a promising proof of concept that this is a good thing to do. How do we translate this deep neurobiological knowledge with this intervention into a vision for the future treatment of intrusive thinking? I just want to close with the following 2 slides. The first is to emphasize that we are in an extraordinarily rare position here. Why? Because what we've done is we've taken a single psychological circumstance in the process. I'm getting you want to stop treatment. We've taken that and we really into it. We characterized it at the level of cognition, at the level of interacting brain systems, at the level of circuitry and neurochemistry. We've dealt with the tools to ultimately understand it at the neural level. Yet now we've taken it and we've connected the dots to mental health intervention. So now we can take this fundamental neurobiology and aim it at improving mental health or transdiagnostically across people. Why should it have such a big mental health impact? And I'm going to share with you here a diagram, which I spent a lot of time on, so I will look at. Well, before that, the idea behind this diagram is that retrieval stopping may be a keystone mental habit. What do I mean by keystone mental habit? I mean that it's a habit that if you adopt it, would have cascading benefits across other areas of your life. So it's a central habit that propagates to other things that you experience. Let me illustrate why I think. Look at that. Okay, this is intrusive thinking. What does intrusive thinking affect? Intrusive thinking causes sleep problems. Everyone in this room knows this. You've all been worrying, cycling through intrusive thoughts. The next day, you have fatigue, low energy, you can't focus on anything. Intrusive thinking causes stress. Chronic stress makes you more vulnerable to illness and makes it slower to recover, increases inflammation. Intrusive thinking causes chronic negative mood, causing someone to feel really overwhelmed. Everybody could easily overwhelmed by everyday conditions. It causes poor focus and concentration and mind wandering, which causes time management. Poor decision making, strained relationships. Anybody who can't let go of whatever's distressing and cycling through their minds and has to process it with everybody around them becomes very unpopular. Let's face it. And so then they're isolated, right? And then reduced performance, depressive rumination. So the thinking here is that if we can intervene here, give people a sense of control over their own thoughts, empower them. Then they have the discretion to think about things or to not think about things. And that will have cascading benefits across this spectrum of symptom domains in mental health. So here's how I propose that we do it. We develop a stratified intervention for enhancing adaptive forgetting. It's composed of an initial universal first-line intervention of suppression training, exactly the kind of thing I just described. It's brief, it's scalable, it's low cost, it's low burden. It can be delivered online or in person. It's short in its duration. And it should have this immediate benefits on intrusive thinking if they know how to do it. And my thinking is that many people who are waiting for treatment, mental health treatment, who don't have the money for mental health treatment, if they have something like this, it will help them significantly. But it won't help everybody. So the people that it doesn't help, that have a poor response or are worsening, I would say that this is basically acting as a filter for distinguishing in a large group of people with intrusive symptoms, those whose thinking derive from treatable behavioral processes, from those who have something neurobiologically malfunctioning. So a deficient prefrontal cortex, deficient connectivity, deficient neurotransmitters. And that previously, without this filter, you would think everybody has those conditions. But this, if you can improve people significantly with this simple intervention, you can probably weed them out as not having neurobiological deficiencies. That leaves these people on whom you can then focus your resources to do things like diagnostic imaging with a retrieval stopping task to develop a second line augmentation of the treatment. So use fMRI to assess activity and connectivity and stopping networks to retrieve stopping, and ultimately develop precision augmentation, mechanism-informed, individualized intervention. So you'll learn if you do this, that some people have something the matter with their prefrontal cortex or with the white matter connectivity to intermediate nodes in the pathways, or with GABA neurochemistry in the hippocampus. These are separable causes. And if you know problem is, you can then target that, for example, with GABA agonists, and then pair that with the training to increase the impact of the training. But this is the stratified intervention dream. So I think then with these three parts together, that this vision helps us to exploit the deep neurobiology that we've developed here, combine it with the intervention to really channel what we're learning about the basic structures and processes of the brain to the benefit of the public. And if you forget everything I spoke about today, remember this, that forgetting isn't a bug. It's a feature.